Dr. Andrew Kaufman. Now, it's pronounced Kaufman. I just want to make sure everybody knows that. I always ask to make sure. So, a lot of times, I've been referring to him as Kaufman for a long time. It's Kaufman. Now, surviving the age of the virus. Well, why is that in quotes? So he's going to tell us about it a little bit. Andrew Kaufman, MD, is a public speaker, researcher, natural healing consultant, and inventor. And he's invented a couple of medical devices. He might tell you about that too. And COVID-19 whistleblower. Whistleblower from what to what? Well, we'll talk about that a little bit. Dr. Andy has a BS from MIT in molecular biology and completed his psychiatric training at Duke University Medical Center after graduating from the Medical University of South Carolina. He spent many years in the medical field and practiced as a forensic psychiatrist and expert witness. But they come after you. Well, of course, they want to take your license away and they want to make you wear a mask. Well, what if you won't wear a mask? How are you an expert witness in court? Feel me? And like, no. So he gives up a lucrative practice, business, and sacrificed his personal financial interest for us. So I really appreciate that. That doesn't happen all the time. When he learned that many of the modern medical practices were harming people and not helping them, he gave up his lucrative medical career and began researching and understanding the relationship between body, mind, and spirit, and how to use nature to heal your body. Dr. Andy's new practice teaches people the vital knowledge to become their own health authority. Now, what they're doing, virtual consultation and so on, but apprenticeships for doctors. As I interview doctors, even naturopaths, nurses, anybody in the medical industry, you know how much time they spend on nutrition? Like none. I had no idea. So who fills that gap? Who's going to do that? Dr. Andrew Kaufman is helping. Dr. Andrew.
Hello, everyone. Gosh, I want to thank all of the anarchists out there for giving me such a warm reception at this event. It's really been a treat, and I've been really impressed with the degree of medical and biological knowledge that you all have. And I know that we recognize that we have experienced an opportunity here during this pandemic, but not only an opportunity, also a necessity. Because if we don't learn about health, how our bodies work, how to heal, then we're in danger. So that's what I'm going to talk about today. And my slides are here. Excellent. So I've talked about this topic a lot, about the existence of a virus, and I'm going to give an overview because of the time constraints today. But I want you to know that there's plenty of material to dive deeper. Once I completely refute and disprove virology, I'll move on to talk about the natural healing supplies and a little bit of instruction about how you can handle virtually any health crisis on your own terms at home. So, why is this the most important question? Does this SARS-CoV-2 virus actually exist? So we're reminded of a little bit of logic and reason. So we look to Carl Sagan. Extraordinary claims require extraordinary evidence. And there has been an extraordinary claim made that an invisible particle is circulating the world and causing the worst illness in 100 years and justifies all of the tyranny that we're facing. Hitchens' razor, a principle of reasoning and philosophy, what is asserted without evidence may be dismissed without evidence. Many people who criticize the work that I've put out there use logic fallacy. These are irrational arguments that subvert a true, genuine discussion and interchange. So I want to draw your attention to a couple because I'm sure you've all experienced having discussions about this issue with people, and often they use these techniques, okay? So, for example, appeal to emotion. We've seen emotion enter into these discussions many times. One that I want to highlight specifically here is burden of proof, because many people put the burden of proof on me when that is erroneous. So I want to make it clear that I am not making any claims and I don't have any theories. And there's a maximum of law that says he who makes a claim has the burden of proof. And the claim has been made by the scientific establishment, by the field of virology, and I am simply examining and refuting the evidence put forth to back up that claim. Of course, I've also faced a lot of ad hominem. You're not a virologist, you're not even a doctor, right? Things like that, which are irrelevant, because each and every one of you, no matter what your credential, no matter what your education, no matter what your background, you're all capable of understanding science, scientific experiments, how to look at information, how to discern what's accurate and what's not. And those are the skills that we need to all be developing now. And I know many of you have already been doing this. But this is really the challenge, and we need not be detracted by naysayers who are trying to derail a genuine interest, a genuine discovery, and the real truth. This article was published 17 years ago. It's one of the most widely cited papers in all of the scientific literature. And yet it seems that we've all forgotten that more than half of all published scientific research findings are false, and provably and demonstrably so. Professor Ioannidis is at Stanford University, a very mainstream and well-respected individual, and has also been critical of the exaggeration of the death and infection numbers from a mathematical point of view, but has not looked at the same material that I have, unfortunately. So I think we all know where on the truth spectrum virology lies. So here are all the things that are all rooted in the existence of a virus. Practically everything that we're facing that is causing us stress in the world right now, dividing us from the ones we love, making it very, very difficult to travel, although we've all done it. So why is it so important? The truth is always at the foremost level of importance. But also, if you have the false belief that viruses are real and cause these scary diseases that could be announced at will, you are going to be vulnerable. Vulnerable to false narratives, vulnerable to giving away your rights and giving away your freedom. So the proof of the existence of viruses, and in particular this virus, rests on three pillars. And I'm going to go through them with you today. The first pillar is the so-called virus isolation, where they change the definition of isolation, and I'll explain that further. Number two, electron microscope images, so not the CGI images that are made by an artist, okay, which are peppered throughout this slideshow, but the real electron microscope images. And three, in silico genome sequencing. And it's not based on the blockchain. Pillar one, isolation. So this is what a proper isolation experiment would look like. And this has been done with other nanoscopic particles, such as exosomes, which come from our own bodies in response to being poisoned or with radiation or stress or cancer. And also for what they refer to as viruses in single-celled organisms like bacteriophages and bacteria, as well as something called giant viruses, which are found in sea algae, one of which was discovered by Dr. Stefan Lanka, who I'll talk about later. So, of course, the animation struggles a little bit, but what you would see here if the animation did work, and hopefully we'll have sound for the next clip, hint, hint, is that you take a sample from a sick person, which is represented in the test tube. Then you'd put it through a filter to remove the bigger things, the debris, the human cells, the bacteria cells, etc. And then it would go up in that test tube here, and you see the different colors represent different concentrations of a sugar solution, sucrose, and it's called a density gradient because the more sucrose is in there, the denser the solution. And this looks just like a B52 if anyone's going to be at the bar later. You spin that at high speed in an ultra centrifuge for the amount of time, and whatever you lay on the top of there, so you lay your filtered sample on the top of that test tube, and as you spin it, it migrates down the tube and gets to the point where it's the same density as whatever is migrating down the tube. So if there are a whole bunch of particles, all of the same size, shape, and density, they will all form a tight band like that dashed line in the green zone there. And that is a purification method. So you can take them out of there with a needle or a pipette and then study them, characterize what they're made of, look at them under a microscope, remove their genetic material, if there is any, sequence it, etc. And that has been done for bacteriophages and exosomes many, many times. In fact, there's even articles published on other methods of purifying those particles that are even fancier and more modern technology, because anyone want to guess how long the centrifuge technique has been around? It's been since the 1940s at least. So it's not exactly beyond our technological abilities. Once you purify the sample, then the microscope image at the bottom, that is what bacteriophages look like. And you can see they have a very distinctive shape with a hexagonal head and then a tail. And you can see that that is the only thing that you see in that image, and all the particles are identical. Now, these little dots and bubbles are just artifact. That's the background artifact because you're at the limit of magnification there. But you see all the real particles are identical, same shape, same size, and that's how you know they're a real thing. And I hope the sound works, but I'll voice dub it if it doesn't. The next slide has Luc Montagnier. How many of you have heard of Luc Montagnier? And he recently passed away. And I, of course, want to be respectful to anyone who with an untimely demise. But he's an important figure because he was awarded the Nobel Prize for the alleged discovery of HIV. And in this interview, by his own criteria, he actually concedes that he did not really make that discovery.
Okay, I think I was taught wrong then. I was taught that the purification part was to get all the proteins from the virus.
No.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
To make sure you have a real virus. So, as we can see from that, you need to purify the virus, as I outlined, in order to show that it's real. Now, how many of you have seen the purification of SARS-CoV-2? Anyone? Have you seen the Freedom of Information requests asking for proof of purified SARS-CoV-2? And as we know, none of those came back with any evidence. So I'll explain what they do instead. So they start with the same biological sample of a sick individual. Wait, let me pause for a second. I forgot that there's an important principle of scientific experiment that you always have to do a control experiment. because you design an experiment to test something in particular, right? Like, is there a virus in the sample? And you want to make sure that the evidence you're looking at means that and doesn't mean something else. So you run the experiment without a virus in the experiment at all and make sure that you get a different result, a negative result, so that then when you add the virus, then you know that the result is from that and not from the experiment itself. And this is how you do that control experiment. Start with the same lung fluid, but from a healthy individual, or perhaps even don't put any biological material at all. But this would be proper. Filter it, purify it, same steps. But then you see there's nothing under the microscope because this is a control experiment. It's not supposed to give you a virus. Now, this is what they do in virology. They start with a biological specimen from a sick person. They filter it the same way. And sometimes they do a little centrifugation, but it's really just to remove big particles. It's not the purification step, so don't be confused. Then they add it to a foreign cell culture. Now, this is quite interesting because what they tell us that viruses do is that they invade our body, and in this case, the lung tissue, and they multiply in the lung, causing damage to the lungs and make people sick and risk of dying. So they're already being cultured in the person who's sick. But then they say that they don't get enough from that, so they culture it in foreign cells. Now, interestingly, they usually use cells from the kidneys of monkeys, not even human, not even lung in this case. But they don't just do that. They also use very unusual conditions for this culture. They deplete the nutrition way down. They add not the normal concentration of antibiotics, but 2 or 3 times the normal concentration. They choose specific antibiotics that happen to be toxic to kidneys when they're using kidney cells in the culture. And they have to add fetal calf serum, which is a source of nutrition, and it has special factors from a fetal animal, just like many of you may have heard about the health benefits of colostrum, right? The first milk that a baby's supposed to drink. There's something special about babies and fetuses, right? We know about fetal stem cells as well. So the fetal calf serum makes sure the cells survive long enough that they can do the experiment, because all those other poisons are enough to kill them right away. Now, when they culture these cells, they first look under the microscope, not an electron microscope, but one at lower magnification, and they look at multiple cells, and that's what this looks like here. But you could see it looks sick. I mean, just looking at it, and those kind of holes in it are called plaques, and that's what's called cytopathic effects. And that is just non-specific cell damage. Okay, so if we have cells and we treat them badly, we'll get cytopathic effects. But this is given as the initial proof of the virus, that because of that cytopathic effect under the microscope, back here, that means there was a virus. Now, did they think there might be something else in the lung fluid that might cause that? Or perhaps something in the experiment? So did they do a control experiment? Well, they don't do any anymore, but they did one in 1954. And this is John Franklin Enders, winner of the Nobel Prize for helping to manufacture and develop the polio vaccine that he's not known for, but that's why he was awarded the prize. And essentially what he did is he took the manufacturing process for the polio vaccine, adapted it to measles, and he didn't say this at the time, but later this became the experiment I just described that is put forth as the proof of the existence of viruses. Now, in his experiment with measles, which was virtually the same procedure, except he added milk to the sample, but he had measles patients, he took their snot, added milk, put them in monkey kidney cells, with depleted nutrition, with antibiotics, got cytopathic effects. But he also did the experiment with no measles virus. And he put it in the tissue culture with the kidney cells and the antibiotics and the nutritional depletion. And guess what? He got the same cytopathic effects. And you can see from these quotes on the top where it's described, and if you look at the top right, the cytopathic effects, which superficially resemble those resulting from infection by the measles agents, may possibly be induced by other viral agents present in the monkey tissue. And in another place that he said it's caused by unknown factors, not very certain. So essentially, this casts major doubt on this experiment because you got the same results with the control. So if you're a scientist, what you would do is go back and design a new experiment. But instead, this has been adopted and repeated hundreds and hundreds of times, each time claiming to find a virus, including in SARS-CoV-2. Now, recently, a virologist who left the academic field of virology, once he learned the truth that I'm telling you today, Dr. Stefan Lanka, in the last year, he has worked with an anonymous academic scientist who's afraid to let anyone know that she did these experiments in her lab. He repeated the control experiments, and this time he didn't put anything in there biological at all. So he had the cell culture, he starved it, he poisoned it, but did not add any biological specimen, and he got the same result, cytopathic effects. And here are the actual slides from the experiment. The top row there is before the experiment began, when they were given full spectrum normal nutrition, no antibiotics, no fetal calf serum. Here, that continued. This is the control of the control. So Dr. Lanka knows about control experiments, and he made sure he did his. So when he did not change the nutrition or add antibiotics, the cultures were normal. The second column was nutritional depletion, but not full. I call it the bikini diet. And you can see that it's not quite as normal as the control, but you wouldn't call it full cytopathic effects. The next column, where they added the antibiotics, that's the real control. And you can see the plaques formed and the cytopathic effects. In the last experiment, they added yeast RNA for a different reason to go on and do genome experiments after that. But you can see that just some RNA from yeast made it more toxic. When you add lung fluid, there is a lot of RNA. So could just the RNA from lung fluid also contribute to these cytopathic effects? Part 2, electron microscopy. Now what they do is, once they identify the cytopathic effects, they then pull the fluid out of that, and they process that to go under the electron microscope. And processing tissue for electron microscope really beats the crap out of the tissue. I mean, they freeze it, thaw it, bombard it with heavy metals, including things like plutonium. They shoot it with an electron beam that vaporizes instantly all the water in the specimen. So essentially what you have in the images is you're only seeing the metal stains. They've replaced all the actual biological molecules. So you have to take this with a little bit of a grain of salt, that some of these things may simply be artifact. But I'm going to put that aside for this discussion. That's for, another day. But here are two images. They both have the same size. That's said to be a coronavirus. They have the same shape. They have the same little dots on the outside that are said to be the spike proteins. So which one is SARS-CoV-2 and which one might be something else? So you can all see this, right? All right. So I want you to vote which one you think has the virus. Is it this one on the left? Raise your hands. All right. 2. How about the one on the right? All right, now I can see that you guys don't really want to raise your hands, but I don't think anyone can really tell the difference, can they? Turns out one is from a kidney biopsy of someone with kidney disease before COVID existed, and the other one is alleged to be SARS-CoV-2. And those images are from this article published the summer before last in Kidney 360, a peer-reviewed mainstream journal of nephrology. And what they noticed is they saw the same exact particles that are said to be coronaviruses in people who had kidney disease that they were studying their biopsies under electron microscopy. And when they saw this, they actually looked it up and found out that in 2003, the CDC said the same thing. Yes, the CDC. So in other words, you can't tell what these are by looking at them. There's in fact no way to tell what they are. I want to mention that there are many other examples of this confusion, but I don't have time to go through them today, but there's certainly things that you can find. So we've essentially disproven 2 of the 3 pillars of virology, and now on to the third, the in silico genome. So give you a little bit of a definition, but essentially what this is that it's inside of a computer. So it's not taking biological material and doing chemistry. That's only a small part of it. It's all a computer putting it together in a variety of ways, and I'll highlight that a little bit. But in other words, it doesn't exist in the real world. So this is the first paper published in Nature, and you can see at a very early date, it was accepted January 28th, 2020. I believe that was before the first isolation experiment was submitted. So they had no real evidence of anything at this point, okay? But this is what all of those variants, all over 7 million, I believe, and counting, are all based on this one study, because this created a template. And then they looked for anything that matched the template, and you could find it almost anywhere. So what did they do in this experiment? Well, how many people you think you'd want to take virus samples from if you were going to sequence a genome? Maybe 1,000, 500? Well, they thought they only needed 1. So 1 person in the world out of the entire world population supplied us with this genome. Okay, once again, no control experiment. They didn't have a healthy person do the same experiment and be unable to produce the genome. Okay, so this mistake is part of the heritage of virology. So they took 1 person's lung fluid, they took out all the RNA. Now, where did those RNA pieces come from? No idea. A lot of them came from the human, but there's also other microorganisms that live in our bodies and in our lungs. And then whatever we breathe in the air before they take the sample is still there. So there's lots and lots of different sources of genetic material, including RNA. They said how much RNA, what are the lengths. They took out all of the longer pieces because they want to do this experiment fast. They need to have a quick turnover so they can get the vaccine manufacturing process going. They make a library which just is PCR again, amplifying everything, so it introduces error. Then they remove some of the sequences arbitrarily, but they don't explain what they removed or how they decided what to remove in their paper, in their published protocols. Then they put everything into a computer, not 1, but 2 software applications. And they had some interesting names, Mega Hit and Trinity. And they each spit out hundreds of thousands of possible genomes. That 56 million number, by the way, that's how many individual little pieces went into the computer to crank out a genome that's only 30,000 long. They were already looking for 30,000 length. They had 56 million fragments, all roughly 100 to 150 bases long. So they took only a few 10,000ths of those pieces. How did the computer know which of those pieces were from the virus? It didn't. So here are the problems. The genetic material is not taken from a virus, it's taken from a person. Not a pure source, not from a single organism, but from multiple organisms. Computer software constructs the genome from an insane number of little pieces, and then it puts out over 1 million possible genomes, but they're all thrown out except for one. And they just picked the longest one. So all the output from one software application were just discarded with no reason. The other one, they took the longest one, they said that's it. But here's a problem. They said what they have doesn't include the ends for some reason. Now, how did the ends not get in there? No idea. So they go back to the original sample and use the PCR trick to make ends. Then they compare it with all the genomes out there in the database, but really they were only looking at coronaviruses. And they found it's 89% similar to a bat SARS-like coronavirus, whose genome was done by the same exact technique. Now, we are about 88% similar to house cats. So do you think 89% similar means they're the same species? I don't think so. But they thought so. And they said it's a SARS-like coronavirus. So here's a compare and contrast chart between a real genome and the in silico genome, because we do have real genome sequences like from humans and animals. And in those, what they do is they take the genetic material from the actual animal that they're sequencing. So if it's a human, they take human cells, not human cells, and bacteria cells, and camel cells, and hippopotamus cells, just human. They can reproduce it without error. There's no human variance, right? We're all humans. We're not Delta humans and Omicron humans. So this represents the actual code of an actual organism, whereas the in silico genome, a mixture of unknown sources, is the starting material, cannot be reproduced without error, which is what the variants are. They're simply inability to reproduce the results. And we have a theoretical code of a theoretical organism. So if, as I said, I believe we can take the if out of here, because I think I have actually disproven the existence of this virus. So, thank you. If there's no virus, there can't be any spike protein from a virus, can there? There can't be genetic mutations or variants. You can't develop a test for something that's not real. And you certainly can't make a vaccine for something that's not real. And you can't determine if something that's not real causes a disease. So once you understand this fundamental truth that the evidence put forth to prove virology can be debunked in less than 40 minutes in Mexico, you will instantly understand that everything else is complete bunk and can go right in the trash. And then we can start building our own life, our own infrastructure on our own terms. This is the recipe that's come out of this pandemic, and you know they're planning to do it again. We've heard rumors, smallpox, Marburg. There's now a rash of a new dangerous HIV virus causing AIDS. A lot of these are simply ways to explain away vaccine injuries, but this is going to continue. So here is the recipe that they've used up until now, which is they do these virus isolation experiments. They make a toxic vaccine from the tissue culture filtrate. That's like the old vaccines, like the one that Enders made for polio, like chickenpox, like meningitis, like the flu, all those. They get FDA approval. It takes 10 years on average. Now we have the new technique, the new recipe. We don't even need the isolation anymore. We're just going to use the computer simulation, because that gives you the code to make the vaccine. And since they declare an emergency, they can get it out without safety testing and effectiveness testing, which takes 10 years or more. So they can just pretend or say, we hope it's safe and effective, or like it says in the EUA letters, that it may possibly theoretically be safe and effective. So we have to have this knowledge to prevent this from happening over and over again, each time chipping away at our freedom, putting us closer to the digital surveillance state. So once you know there are no viruses and no disease caused by viruses, and you know that if you go into a hospital or a medical facility, they're going to be testing you, diagnosing you, ventilating you, sedating you, giving you toxic poisons. So you don't want to do that. So how do you handle things on your own? Well, I'll tell you, the best thing is start getting healthy now so that your chance of an emergency is really low. I did a workshop yesterday that is the kind of thing that can get you started, and there are many other health experts here who also have this knowledge. And I urge you to start thinking about taking care of your health on your own, learning what you need to know. So I'm going to focus on emergencies and some of them that you may not have any control over, like accidents today. So this is for trauma, cuts, scrapes, broken bones, lacerations, puncture wounds, etc. Buy some aloe plants, grow them in your house, water them, take care of them. Thank them for being there to help you heal when you need them. The way that I handle lacerations, and I have kids, so I'm dealing with this all the time, is quite simple. You rinse it off with clean water. I usually use RO water. Distilled water is good. If you have a pure source of spring water, just clean off the debris. Don't use soaps, abrasives, iodine, all that poisonous stuff. You don't need it. Now, I do sometimes, if it's a bad one, or if it looks like it's starting to get infected, put a few drops of turpentine on the skin around the margins of the wound. That will help dissolve any toxins that may have been contaminating from the wound itself, or anything your immune system brings there to try to purge through this cut that would cause like an infection or an abscess type of situation, which is really your body trying to purge toxins. And then simply cover it with an aloe leaf. So you cut off a piece of the aloe leaf, split it open so the slimy side goes right on the wound. And that performs like a magical matrix of healing. You get amazing results. Then you put some bandage material. I like the self-adhesive bandage material. So there's no tape or adhesive on the skin. And just wrap it up, put some gauze right over the aloe to absorb the moisture, and just leave it for 24 hours. Change it once a day. You'll see that the wounds will heal rapidly and you'll get excellent cosmetic results. Even wounds that you'd think need stitches will give you better healing cosmetic results with this technique. So I only reserve stitches for unique situations if you have to attach deeper tissues together. And that requires a little bit more instruction than today. But you might keep some suture material. And if you just have 100% cotton thread and some good needles, that will work as well. But you can buy veterinary, quote unquote, suture material because you're not a doctor, but it's the same exact thing. So don't hesitate. It's available online at many sources. It's good to have a variety of materials for splinting and making soft casts, and these are just to immobilize injuries like ankle, elbow, things like that, where you might have trauma or especially breaks. These things are readily available at medical supply stores. I'm not saying to go out and get fitted for every joint in your body and keep that on hand, but there are a few general devices, and if you learn the principles of immobilizing a broken bone, it's not very difficult. You definitely need to be prepared to use a tourniquet and possibly do an amputation, which may not be the fun thing that you want to hear about, but I'm talking about, wilderness survival where you don't have access to other sources. And in any kind of trauma situation, the most important thing to do right away, other than if there's breathing or problems, is to stop bleeding. That is what risks the life. And that's even true with something like a gunshot wound, that if you don't stop bleeding right away and try to go to the hospital, your chances are much worse. And they have nothing magical that they do to stop bleeding at the hospital. Like, they don't have cut you open to stop the bleeding. If they didn't stop it before they cut you open, you wouldn't survive the surgery. So you need to learn the principles of dealing with bleeding situations, and that includes a tourniquet, which you can, tie a string extremely tight. There are specific ways to secure it, and there are even ones you could buy that have the built-in mechanism. But if you have a bandana and a pencil, you can make a tourniquet, no problem. Turpentine, the most amazing healing solvent. If your family member or you or your loved one looks like they're about to die of a serious illness like sepsis or heart attack or something like that, if you give turpentine and castor oil, you may just turn the whole thing around. First, do water, rehydrate, then that's the next step. And you can have amazing results in those really scary situations. You need to have clean water. I recommend getting a reverse osmosis system or a distillation system unless you have access to primary water from a spring. And hopefully some of you do have that. Let's talk about binders and baths. So these are like chelating agents, as another word for binders. And baths can be very healing because they detoxify you. And things like clay and Epsom salts are good in baths. They will pull toxins out through your skin. They can help resolve fevers, as can enemas. The activated charcoal, I brought some with me here just in case. But certainly if you have any food poisoning or diarrhea illness, activated charcoal can generally knock it out quite quickly. And also it's useful in a poisoning situation if the substance that's poisonous is still in the gut, or if it's on the skin too, you can do it, but I'm going to talk about that separately. So, bites and poisoning. Bentonite clay is an amazing substance for any type of bites on the skin, jellyfish, bee stings. You get sprayed with a skunk. If you put the clay on the area, sooner the better, it will suck out the poison. It works amazingly well. Some of the other binders also may work well for that. If there's a poisonous ingestion, like a child accidentally swallows a cleaning solvent, or perhaps someone's pharmaceuticals that they forgot to dispose of properly, you can survive almost any poisoning with high-dose vitamin C, milk thistle, and NAC. NAC is a source of sulfur, which your liver uses to make more glutathione, which is a potent antioxidant to help detoxify what's in your blood and so that your body can excrete it. Milk thistle works the same way, but on a slightly different system of conjugation. Very powerful stuff. So like if you have poison mushrooms, Tylenol overdose, milk thistle alone can save your life from those conditions. And then we have vitamin C. So there are studies with animals that show they give one group of animals a lethal level of formaldehyde that's used to embalm cadavers, give the other group vitamin C and the same exact amount of formaldehyde, and guess what? The vitamin C group lives, and they all die in the other group. So this is very, very important. It needs to be high doses. So if you have ascorbic acid, I would recommend something like doing a bowel tolerance where you take 2,000 milligrams every 15 minutes until you get some diarrhea. And then that is your daily dose, essentially, and you can keep taking it at every 2-hour intervals or 4-hour intervals after that until you're completely recovered. Very, very effective stuff. Healing solvents, as I mentioned. So castor oil here. Castor oil is very, very useful, not always internally. I save that for life-threatening emergencies, but you would take a quarter cup of castor oil with 1 teaspoon of pure gum spirits of turpentine. It has to say that. Otherwise, it has poison chemicals added to it. And these 2 things will make a huge difference in a life-threatening situation, but they have many other uses, and they're excellent to use on the skin. So mosquito bites, skin infections, etc. DMSO is another solvent that we'll be talking about soon in one of my webinars upcoming.
Excuse me, Dr. Kaufman. We're already 10 minutes past. We want to keep them going a little bit, right? I mean, how much
Well,
longer?
thank you. This is my last slide, actually. Now you tell me.
Well, now's a good, well, like it made any difference. All right. This is tomorrow at the yoga space. They set a place from 11:30 to 12:30 for a Q&A with Andrew. All right. So tomorrow morning, 11:30 to 12:30, if you got anything that you want to ask, we're going to have that opportunity. Go ahead and wrap it up because I know we want to get the NFT auction. They love on some Andrew. So go ahead, and you need what? 3 minutes?
2 minutes.
2 minutes. Go.
So if anyone attended my workshop yesterday, they know I love to talk about enemas. So I can't leave it out, of course, but this is a really, really potent technique to get bad things out of your body. In the past, this was the primary treatment for pneumonia, for sepsis. It's cured psychotic depression, schizophrenia, all kinds of things. You have to go back to the 1930s to find articles because there's no profit in it. But everyone should have an enema kit in their home for those situations. I can't emphasize that enough. It's good to practice so you know what you're doing if the time comes that you need it, but it's actually can be a useful regular practice. Also, having some laxatives, like those vitality capsules, are a combination of herbs like senna, cascara, barberry root. It also has things like ginger and garlic in there, which help with the comfort level. But these types of things, if you're sick and you do enemas and you poop, you will get better. And I just want to announce everyone that my brand new film Terrain, which just recently premiered, thank you, which goes into more detail and is not just about a geeky science movie. It covers the human story too. But after Anarchapulco is over, we're having a special replay this weekend, so everyone here can watch it free with this link, and I hope you guys check it out and look into this issue a little bit deeper. Thank you so much for inviting me here.
There was a time when a pirate was free to make his own way in the world. That time is coming to an end. Navigating along the 7 seas and 4 corners of the earth for innovation and solutions that bring you true financial freedom. It's been 12 years in the making, perfecting the art of decentralization and privacy. In a world where privacy is a luxury of the past, where every transaction is being monitored by a centralized entity, and true anonymity seems like a fairy tale. Pirate Chain came to change the game, the first cryptocurrency to bring you the highest anon set ever. Unlike the rest of the virtual coins out there, where privacy is not a main feature. Pirate Chain's goal is to protect its users with the most sophisticated and most acclaimed privacy protocol in the world of cryptocurrencies. Are you going to get on this ship, or are you going to keep putting your financial freedom at risk? Visit us at pirate.black, because financial privacy today is freedom tomorrow.
Introducing a powerful dietary supplement, over 1 million years in development. A highly concentrated, complex, natural formula, rich in trace minerals as well as fulvic and humic acids. Mother Nature's ancient elixir for energy, revitalization, and long life. Shiljit Mumio means conqueror of mountains, destroyer of weakness. Take in the nutritional wisdom of the ancient past. Unleash the vibrant, healthy being inside. Become the conqueror.